is a scholar on ecology and evolution of plant-animal interactions with a particular focus on pollination. He is currently director of the Center for Functional Biodiversity at the University of KwaZulu-Natal, in South Africa. And he also held the South African National Research Foundation Chair for Evolutionary Biology from 2007 to 2022. Formerly, he got a PhD in botany at the University of Cape Town in 1994. And then he had two postdoctoral leaves abroad at the University of Haifa in Israel and Uppsala University in Sweden. Then he came back to South Africa, to Cape Town again, for a third postdoc, the Smooth Fellowship, that is prestigious in South Africa. And then after that, he moved to the University of KwaZulu-Natal, where he has remained, as far as I know, since that time. And as a full professor since 2007. He has published more than 350 scientific papers and 4 books. And his editorial services include, or have included, being in the editorial board of several prestigious journals, such as Ecologia, Arthropod-Plant Interactions, or Proceeding of the Royal Society B, among others. More recently, he has opened his focus by including chemical ecology of plant-animal interactions, with a particular interest on the analysis of volatile emissions from plants and how they affect their interaction with pollinators. His empirical works and reviews on pollination biology, ecology, and evolution remains as the most cited in these topics across the world. Personally, I will stress the point that Professor D.Jhonson, together some other colleagues in South Africa, has changed our views about specialized pollination, at least in terms of morphological and functional fit between flowers and pollinators, which we thought that it was exclusive of tropical regions, if any. Now we know that there is a small but meaningful region in the world, the Cape, in its widest sense, even so it's quite narrow, which is Mediterranean, non-tropical, and support a rich flora where is very frequently found specialized pollination. In a sense, the Cape is not only a biogeographic region, that we know from the late 19th century, but also is a key place for pollination biology. If there is any biogeography of pollination biology, it should stand as a very particular region. And Steve has some responsibilities for that. His own personal thoughts about these topics are nicely expressed in this. Sorry. Can you put the slide there? Oh, okay, thank you. His own personal thoughts about these topics are very well and nicely expressed in this interview, showing this slide, which I strongly recommend. I think that's enough for now. Steve, we're eager to listen to you.
Thanks, Juan, and good morning, everyone. I'm going to give you a talk about my research on floral divergence among populations, giving some examples, but bearing in mind that not everyone works on flowers, I will try at the end to make some more general points about the study of evolution in plants. So what I'm going to talk about first of all is what do I mean by a pollinator niche, and how are pollinator niches associated with floral trait syndromes. And I want to propose a different way of looking at floral syndromes than what has been the case traditionally in the past. I want to look back at the concept of ecotypes, which is about 100 years old, and see how it can be applied to floral evolution. And then I'm going to give a number of examples, mainly from my own work, for two reasons. One is I know that work the best, and secondly, because I think it's a nice way of showing people some of the diversity that we have in Southern Africa. And I want to end with some of the challenges we face in working on this topic. So, as most of you would know, Darwin attributed the diversification of life to natural selection. One of the examples used was finches in the Galapagos. And for an evolutionary biologist, a niche is basically an ecological opportunity that can be exploited if a plant or animal has the right set of traits. And so a theme in the natural world is specialization of plants and animals, not to differing degrees, but the reason for specialization primarily is that a plant or an animal cannot be a jack of all trades. You cannot have a bill morphology, for example, which allows you to feed on any kind of food. And that is true for whatever set of traits one is looking at. And so trade-offs are a very important theme in the work that I will be talking about. And from a point of view of adaptive radiation, shifts between niches drives trait diversification and can also promote reproductive isolation. So I'm going to talk a lot about geographical variation in pollinator assemblages spatially. Vern Grant referred to this as the pollinator climate. And pollinators are heterogeneous across geographical regions, across biomes, across vegetation types, along elevational gradients, throughout seasons. And it's a kind of a hidden aspect of the niche diversity in natural habitats that most of us are unaware of, but which is incredibly important for driving plant evolution. In fact, pollinators can be part of the fundamental niche of plants. In other words, the basic conditions required for establishment and persistence of populations. We've known for a long time that plants with specialized pollination systems are not easily able to colonize new regions, whereas plants that have, for example, uniparental reproduction that are sort of free of a dependence on pollinators make better colonizers. So this very obvious fact tells us that pollinators are part of the fundamental niche of plants. So to talk about my own history, going back 30 years as a postgraduate student of the University of Cape Town, studying this remarkable butterfly, and it's the sole pollinator of this spectacular orchid, I noticed in my fieldwork that the orchid had more than 80% pollination success when it grew in rocky habitats, but very low pollination success in open habitats. And the first article I ever published was on habitat-dependent pollination success. And this set up a lifelong interest in spatial variation in pollinator availability and how this affects plants. Around the same time, I worked on this interesting gradient here between the flats around Cape Town and Table Mountain itself, which have completely different assemblages of long-tongued fly pollinators, and noticed variation in the flower depth of orchids and some irises. So there's this almost twofold change in the depth of the flowers, which are very long on the sand plain and then shorter in the mountains, where the flies have shorter proboscides. And this has been worked on by others as well, and the pattern has been confirmed as an important gradient in the Western Cape. So I started to see this kind of spatial sort of temporal mosaic of pollinator availability as being something like, for example, the mosaic of soil types that affect plant evolution. So long proboscis flies are an important pollinator group in South Africa, and here you can see how they set up this mosaic. So a plant that expands its distribution range into new regions will encounter new sets of flies, and floral traits often become adapted to new pollinators. Some of them are very local, found only in forest patches. This is the one found only on the sand plain. And if you look at the dates there, you'll see that these flies are not only available in particular areas, but they're also only available in particular times of the year, which is also important. Another very specialized group of pollinators in South Africa involves plants adapted to oil bees, and these oil bees often have very restricted ranges. There are many plants pollinated only by a single species of oil bee, and as these plants expand their range, they shift between different oil bees, which has implications for the evolution of floral traits. And it was shown by Kim Steiner in his work on oil bee pollination in South Africa that the oil bees themselves, because they use their long front legs to gather oil out of flowers, the legs themselves are often highly variable, and you can have a 2- or 3-fold variation in the length of these legs, which is mirrored by the depth of the flowers that they pollinated. So you end up with this relationship over here. There's the 2 entrances where the legs fit into the flower. So going back about 40 years, Mary Arroyo, working in Chile, did some very nice work showing how pollinators vary along elevational gradients in the Andes. Here's some work. I've added some color to this rather scruffy graph from an old photocopy. But you can see here the decline in fly availability as you go up with altitude, and a change in, and an increase in flyability and a decrease in bee availability. And although the bars are smaller, there's an increase in hummingbirds at higher elevation as well. Robert Cruden, also working in Mexico, showed an interesting pattern of declining pollination success in Caliandra species along elevational gradients. And he looked at the number of hawk moth visits in different evenings according to temperature, and showed that the rate limiting step for hawk moth activity at elevation was temperature. If the nights were below 10 degrees, hawk moths basically weren't able to fly. And so any plant that expands its range to elevations higher than those that are suitable for hawk moths may shift to other pollinators, for example, flies. Now, I thought I should show an example from Europe. A very nice example of a ecological gradient in pollinator availability in Europe is the local availability of a group of scarab beetles that are strongly attracted to red flowers. And it's been shown that poppies that have expanded their range into Central Europe have different spectral reflectance. So where the beetles are absent, the flowers are UV reflecting. Where the beetles are present, the flowers are UV absorbing. And here's some work on Mount Olympus in Greece, showing as the beetle availability declines with elevation, the lower populations of this anemone are entirely red, and at higher elevations, the populations are made up of an increasing fraction of pink and purple flowers. Here's an example of another elevational gradient from work done by Hans Jacquemain on Reunion Island in the Indian Ocean, showing that at higher elevations, the percentage of orchids that are self-pollinating reaches as much as 80%. So as you get into this cloud zone, orchids are not as dependent on pollinators. So working with Carl Duffy during his time as a postdoc in my lab, we did some niche modeling, including pollinator distributions as a component of the niche models for plants, and showed that the spatial distribution of plants can be better predicted in many cases if one includes pollinator distributions as part of the model. So in this diagram here, the blue is the purely climatic soil-based prediction, and the green one is the one which includes the pollinators. So for these three examples here, the distribution is more accurately predicted if you include pollinators in the distribution. So let's take a look at floral trait syndromes. Pollination syndromes, as they're historically called, have been referred to. I see these basically as integrated sets of floral traits that allow plants to exploit particular ecological opportunities, in this case a pollinator niche. And the evidence comes from trait-environment correlations and patterns of convergent evolution. So flowers evolve primarily through sexual selection, just like the sexual ornaments of animals, but with some very important differences. Sexual selection in animals often follows a sort of Fisherian runaway process, where the trigger is really some fetish in the brain of a male animal inherited in the offspring, and the female choice inherited by the daughters. In the case of animals, sexual selection can operate through pure male competition, for example, but it often involves shifts between ecological niches. And this is a very important difference in sexual selection between plants and animals, that sexual selection in plants is often correlated with particular habitats in a way that is quite rare for sexual selection in animals. So in both cases, adaptive radiations are characterized by this diversification of the sexual ornaments of plants and animals. That's birds at the top and the orchid genus Diza that I've worked on at the bottom. So these sexual displays show convergent evolution according to particular ecological niches. These are bird pollinated flowers on your left and moth pollinated flowers on my right, illustrated in Stefan Vogel's classic 1954 book, based on a year that he spent in South Africa. So one of the nice things about studying pollination biology is that one can correlate traits with the environment in a way that is much easier to do than is in the case, for example, if you work on leaves. It's true that plant life form is often easily connected with particular environments, but floral traits, the function and functional significance of floral traits is much easier to ascertain than, for example, the leaves that you see at the top there. So at a macroevolutionary scale, many plant lineages are characterized by numerous shifts between different pollination niches. This is the South African genus Laperosia. So there have been at least 17 pollinator shifts, more than the number of substrate shifts in this group. And in the 9 species pairs, all of them show divergence in pollination systems, that should be systems, not syndromes, while 4 pairs present different substrate types. So if we look back at history, the concept of ecotypes, in fact, the term ecotypes was coined by Jutta Turesson, working in Sweden in the 1920s. Interestingly, and I had a discussion with Spencer Barrett about the term, we were wondering if it had infiltrated zoology. And it's interesting, if you look at the literature, the concept of ecotypes is now well established in zoology and widely used, even though it was first used by botanists. And then later in the 1940s and 50s, Jens Clausen started doing essentially reciprocal translocations between different elevations in the Sierra Nevada to look at local adaptation. So Turesson used common garden experiments to establish that there was a genetic base to floral traits, particularly in response to climate, and Jens Clausen pioneered the use of these reciprocal translocations. The theory for pollinator-driven evolution was essentially developed by 2 botanists working mainly throughout the second half of the 20th century, Vern Grant and Leonard Stebbins. And Stebbins made a comment that's very pertinent to this talk, arguing that in the very early stages of adaptive radiation, we can study it as a process rather than a historical event. And Grant advocated the study of different forms within a species as a way of looking at the processes that drive adaptive radiation. He also wrote a lot about what he called the pollinator climate, the spatio sort of geographical mosaic of pollinator availability. And throughout his life, also emphasized the role of reproductive isolation as a sort of an emergent byproduct of divergence driven by pollinators. And then Stebbins, who generally wrote less about pollination than Grant, but wrote a very important review in the early 1970s, where he outlined 5 key principles for pollinator shift that I've found incredibly useful throughout my work. And as we look through the examples, you will see how these principles manifest. The most effective pollinator principle, the idea of character syndromes, selection along lines of least resistance. So if you have a shift, it's not random how the shift happens. It's often related to a pollinator that can exploit a similar kind of flower, that there's often a stage of double functions, that traits serve a role in both systems, and there's evolutionary reversals, but not back to the same position that it was before. So I suggested that the sort of general model of pollinator-driven speciation should be called the Grant-Stebbins model. And I did this in a book chapter published in 2006. So let's look at some examples so we can illustrate these concepts that I've outlined so far. Thought it would be good to start with a European example that I know from my time in Sweden. So Platanthera bifolia is a common European orchid that consists of 2 morphotypes, a short-spurred one that occurs in open meadows and a longer-spurred one that occurs in woodland. And the longest-spurred woodland one is pollinated by this forest moth, Sphinx ligustri, and the short-spurred one is pollinated by a bunch of shorter-tongued moths. So it looks like a nice example of ecotypes. This is a result from a Swedish student's reciprocal translocations between open and forest sites. And for those of you who know studies of local adaptation and reciprocal translocation experiments, this is not a very spectacular result, not a very good phenotype by environment interaction. And I want you to bear this in mind because unlike studying soil or edaphic adaptation in plants, looking at floral adaptation to local environments is fraught with problems. You can't just uproot a plant, for example, endangered orchid planted in a new place. And even if you did so, you're now presenting a rare morph into an environment that has a much more abundant morph. So your introduced plant is essentially suffering from some kind of a Lee effect where it's unfamiliar to local pollinators and is outnumbered by the local form in the new habitat. So it's very difficult to do reciprocal translocations with plants to test for local adaptation in flowers, and I might, if there's time, come back to some of those problems. So what's remarkable is this very small spatial scale over which some of this floral divergence can occur. This is a system that I worked on a few years ago with a postdoc in the lab. 2 different forms of an Erica species in the Cape. They share an immediate common ancestor, in case you're doubtful, because they look quite different. The short-spurred one is on that mountain, short-tubed one. The long-tubed one is on this mountain. There's a long-tongued fly on this mountain and a short-tongued fly on that mountain, and you've had this divergence. Over just a few kilometers, you could probably fly a drone from one side to the other if you had a good battery. So, and this is a theme that I will return to again and again, this spatial scale. These very narrow endemic distributions of some of these key pollinators. There's a system in the east of South Africa, Gladiolus longicolus, an irid, that has 2 morphs. You can see the bimodal distribution over there. A short morph with about short tube morph, about 4 centimeters, pollinated by small hawk moths, and a morph with tubes up to 10 centimeters, pollinated by much larger hawk moths. But the larger hawk moths will occasionally also visit the short-tubed form where the 2 co-occur. Now, the long-tongued moth is found very much in the eastern part of South Africa. This is the density of plants pollinated by this moth across the subcontinent. And if you look at the distribution of the 2 morphs of the orchid, the long-tubed morph is found in the subtropical east, and the short tube morph is found further to the west, where the longer-tongued moth is rare, relatively rare. There's a zone of contact between the two, and this is where it gets interesting, because lower down on this mountain, Mount Gilboa, close to my university, the two forms essentially have hybridized into a single kind of mixture. And a couple of years ago, a postdoc from Vincent Savalainen's lab developed some markers, and we looked at paternity analysis in the population, and there's no assortative mating to speak of between the long and the shorts in this mixed population. Perhaps a little bit at low densities. But strangely enough, about 2 kilometers away on the summit of the mountain, the two forms coexist with slightly separate flowering times, and they maintain their integrity. Now, this issue of how much hybridization is a problem for considering, for example, ecotypes to be species or just ecotypes is a complex problem, and it can't be studied at a single site. The two forms also differ in their scent chemistry. The long-tube one is has scent dominated by the compound linalool, and the short-tube one has scent dominated by osmine, and in the hybrid zone you get a mixture. And in fact, since this graph was made, I filled in many sites here, and this pattern holds across the distribution. And here in the hybrid zone, you can see that the hybrids have scent which is somewhat intermediate between the two. The reason we were interested in this, by the way, is because if there's a scent difference, it could form the basis for assortative mating. The moths could smell, the long-tongued moths could smell the longer tube flowers which have more nectar, and the short-tongued moths which can't reach the nectar in the long-tube ones, if they could smell the difference, they would forage mainly on the short-tube one. So we weren't doing it just for fun. So here's another spectacular example, this time from the Western Cape. This is Erica plucanetti, and you have to be a taxonomist to believe that these belong to the same species. And this was done by someone very experienced in Erica taxonomy, and molecular data confirmed that these three forms here share an immediate common ancestor, despite being so divergent in their floral traits. And the story, which has been worked on extensively by my colleague Timo van den Nit, is that the intermediate tube form is pollinated by this very important orange-breasted sunbird. It's a short-beaked sunbird that's throughout the Western Cape, but not found in the north. The short white flowers are pollinated by moths, and then in two places here, long-tube ones are pollinated by a different, longer-billed sunbird, the malachite sunbird. So the short moth one is derived from the medium-length orange-breasted sunbird pollinated form, but interestingly, the very long-tubed one, pollinated by malachite sunbirds, has evolved twice, once closer to Cape Town and the other one in the far north where the orange-breasted sunbird doesn't occur. This is a very rare example. You know, it's common for people to look at edaphic ecotypes to find examples of repeated evolution. It's very unusual for floral ecotypes to find a pattern like this. I want to look at the example that I illustrated in the title slide. It's interesting for several reasons. This is an orchid Dizoperecta, which has a pink form, only recently discovered in the mountains in the Southern Cape, which is pollinated by long proboscis flies, and an orange form further to the east that is pollinated by butterflies. So Bruce Anderson and I, and a then student of Bruce's, we spent a few days camping in the Swartberg Mountains and documented pollination of the pink form by this long proboscis fly called Prasica longipennis. But the orange form is interesting. It occurs only with pokers, Nepophia species, poker species, which provide a nectar reward for the butterfly, but the orange form is deceptive and relies on mistake visits by the butterfly, which is conditioned on the poker. And it's interesting the difference in the orientation of the spurs. So the orange form has these vertical spurs, very similar to the vertical flowers of the poker. So that's how it looks spatially. This narrow endemic fly pollinates this very local pink form, and then throughout the east, the orange form pollinated by this butterfly. So we were interested in looking at pollinator preferences. So we cut a few flowers with a permit, I think. Put them in vases, set up pairs, and we looked at pollinator choices. In the area where the pink form occurs, the flies had an exclusive preference for the pink form. The butterfly also preferred the pink form. And then in the area, in the orange sites, it was only one where we could get co-flowering form, so we only did it at one site. The butterfly preferred the orange form. But we wanted to know whether this was because of color or perhaps it was because of morphology. And so we used artificial flowers to isolate color as the only trait of interest. So this is what entomologists refer to as arts and crafts research. This is flowers made out of overhead transparency, painted pink, and they fool the flies literally cannot tell the difference between the artificial flowers and the real ones. And the same pattern again. The fly exclusively visited the pink ones. The butterfly shows a kind of a conditioned preference, visiting pink in these pink flowered communities, and then strongly preferring orange in the sites where the poker flowers occur. So providing a good explanation for the color divergence in this plant species. And just for interest, although we haven't really confirmed the functional significance experimentally, this is the difference in spur angle between the pink and the orange forms of the orchid. So here's another example of color divergence involving an iris species this time. I started this work with Lawrence Harder from Canada when he spent a sabbatical in my lab. And because he could only do one season, we then had a student who continued the work as a PhD, and Ruth Kozian, and she did an excellent job over the years in documenting the system. So there's a red form pollinated by this butterfly, which is, you can see I have a particular interest in. It seems to feature in all these systems. And a red form pollinated by the butterfly and a pink form pollinated by this long proboscis fly, which occurs in the Drakensberg Mountains, called Brassica ganglobaurea. So this is the spatial distribution. The pink form is localized in the high altitude parts of the Drakensberg. The red form is much more widespread. And there's an interesting difference as well in the width of the tepals. The pink form has these narrow tepals, and the red form has broader tepals. These traits are completely genetically fixed. So you can't see it very well. There's a dotted line and a solid line on the left. That's the color of the plants in the natural habitat and the plants that are cultivated. These are measures of color, repeatable in cultivated plants, and these are morphological traits. In fact, the only trait that didn't kind of reproduce in a common garden, interestingly, was flower orientation. And it's perhaps because the shade house affects the lighting, the development of the flowers. We're not sure why that is the case, but there is a difference in flower angle that doesn't seem to have a strong genetic basis. So the flower visitors, we've looked at about 15 sites. The red ones are strongly associated with pollination by butterflies, and the pink ones are strongly associated with butterflies as well as by a day-flying hawk moth. But if you look at the effectiveness, the day-flying hawk moth is in fact not a particularly effective pollinator. So you can discount the day-flying hawk moth as an important pollinator in this system. So going back to Mary Arroyo and elevational gradients, one of the drivers here is that the long proboscis fly is found at higher elevations in the Drakensberg Mountains, never found in the lowlands, whereas the butterfly is found in the foothills, in the lower parts of the mountains. You can see the difference here. And you can see that shift in the elevation of the two morphs as well. So we did these translocation experiments, 2 matched sites between the north and the south. And these pollinators kept their strong preferences for pink, the flies preferring pink and the butterfly preferring red. And here the visitation is quite markedly different. in the sites where the butterflies are common in the south, the red form gets the most visits, and in the northern Drakensberg, the pink form gets the most visits. And here's a phenotype by environment interaction experiment that kind of actually works. So the red form outperforms the pink form significantly in the red part of the distribution, and the pink form outperforms the red form in the other part of the distribution. So you have this strong fitness advantage, home site advantage for these color forms. So going on to some relatively weird examples. This is an extremely obscure family, or it's become less obscure. It's a couple of other genera have been added to this family, the Acaraceae. But we worked on a plant called Guthria capensis, known as the hidden flower, because you can walk right past the plant and not see the flowers. And Ruth Kozian and Sandy Steenhuisen, who's here somewhere, and Timo. First of all, we thought this plant might be rodent pollinated because the flowers are so obscure, so hidden away. But after a couple of years of quite intensive camera trapping, the exclusive pollinators of this plant were these large lizards. The plant is monoecious, and the lizard's snout fits very nicely into the morphology of the flower. Now, Guthria, in fact, has a disjunct distribution. So there's a plant hidden away, with the flowers hidden away under the leaves in the Drakensberg, but there's some disjunct populations in the middle of the Karoo. There's some mountains in the arid region of South Africa. And Ruth and Timo went there over the last couple of years, expecting to find the same lizards that we had documented in Drakensberg, and the situation was quite different. Excuse the lack of italics. This is free video editing software. But you can see the pollinated assemblage was quite different, and it involved small ground-dwelling mammals, kind of much like our original expectation, in fact, including dormice and elephant shrews. So quite an exciting and unexpected discovery. So we have there's a very profound difference in the scent chemistry between the Drakensberg and the Karoo populations, but both of them retain this strong orange kind of advertising signal, which is definitely attractive to lizards in choice experiments. So the lizards are attracted both to the orange color as well as to the scent of the flowers. So they seem to play a functional role and lizards, of course, the males are highly colored, so you'd expect a color response in a lizard is not an unexpected finding. Now, the interesting thing about the Karoo populations is that the Drakensberg populations have very bitter nectar. The Karoo ones don't. And the dormice seem to prefer the sweet-tasting nectar, whereas the lizards don't mind the bitter-tasting nectar in the Drakensberg. So on the theme of unexpected findings, and I don't know if Jeff Ollerton is here, but he would like this example because he has a pollinator that's not expected from the apparent floral syndrome. So we had a student from Mexico working in our lab for a couple of years who was working on a whole lot of morphotypes of this orchid, Satyrium longicorda, which is pollinated by hawk moths. And he came back to the lab one day and said he had seen bees pollinating one of these long-spurred ones. And I was extremely skeptical and went out into the field. And it turned out that one of these long-spurred morphotypes was pollinated by oil bees. And they were clearly collecting oil from the flowers. And we documented this over 2 seasons. And this oil form is derived within this clade of these morphotypes of Satyrium longicorda. If you look at the removal of pollinaria from flowers during the day and the night, all the other ones have their pollinaria removed during the night, but this form over here has the pollinaria removed during the day. And if you shorten the spur of the moth ones, pollination success declines dramatically. But if you shorten the spur of the bee pollinated form, there's no effect on pollination success. So this is a recent shift to pollination by oil bees, where the spur is essentially a vestigial trait. It's there as a hangover from the moth-pollinated past. And in fact, it actually makes a lot of sense because Citurium is one of the few orchids that has twin spurs, and we think that ancestrally it probably accommodated the long legs of oil bees, and there is in fact oil bee pollination right at the base of the Citurium clade. Now, flowers can, of course, diverge even without shifts in pollinators. Here's an example of Corteria diffusa, well documented now by Alan Ellis. And he has shown that some of these spectacular morphotypes elicit sexual responses in this small bee fly in the genus Megapalpus, whereas other forms elicit feeding responses. So the same pollinator, plants can exploit different behaviors of the same pollinators at different sites. This is well illustrated in the case of mimicry in plants, where the pollinator can be conditioned to different colors in different parts of the distribution range, as you saw in a previous example. And in this orchid, Dizophryginia, this again has profound effects on fitness. So here's a reciprocal translocation experiment, again with clear-cut results, where the orange form in the area where there's orange nectar-producing plants outperforms the red form, and the red form outperforms the orange form in the west of the distribution, where the nectar plants for the butterfly are red. So you can have the same pollinator exerting different selective pressures in different parts of its distribution range. So this is another wonderful a bit of biology in the Drakensberg Mountains in South Africa. This is the fly Proscica gangobaria and its main nectar plant in the Scrophulariaceae, a plant called Zaleuzianskia microsiphon. And the fly depends largely on this plant for its nectar. And Bruce Anderson and I documented strong covariation in the traits of the flower, the depth of the flower, and the tongue length of the flies across the distribution range in the Drakensberg. And because the fly depended strongly on the scroff for its nectar, we stuck our necks out and used the word coevolution, which I use only under special circumstances, generally. So this is the correlation between the fly and the Zaleuzianskia. But there's a deceptive orchid, which is also pollinated by this fly. And of course, being deceptive cannot influence the evolution of the fly proboscis length, but it kind of tracks this coevolution between the fly and the Zaleuzianskia. So this is kind of going along for the ride. So we applied the term trait tracking to explain this relationship. And then later on, Bruce documented extensive covariation between communities, in communities of plants, between flower depth and the fly at various sites across the Drakensberg. So the filled black circles are the fly proboscis, and you can see how the community of plants pollinated by this fly kind of mirrors this changing proboscis length. And this can occur even at small spatial scale. So here's a ridge at a place called the Sentinel in the Drakensberg Mountains. And over just a span of about 2 kilometers in elevational gradient, you can find a 25% decline in the tube length of the Zaleuzianskia as you go to increasing elevation. And this is matched by a decline in the proboscis length of the fly. Now, being an increase in elevation and a decline in flower tube length, one of course would be worried about phenotypic plasticity. But these traits in the plants are reproducible in a greenhouse. So despite the small spatial gradient, the short tube flowers from high up grow out in the greenhouse as short tubed, and the longer tube flowers from lower down grow out as longer tubed as well. And a student from Erin Tripp's lab visited a couple of years ago and did a reciprocal translocation experiment. And although we found phenotype by environment interactions, this significant interaction term, you can see again there isn't any real significant local morph is not significantly outperforming the foreign morph at any of these sites. So this is a slightly ambiguous result. It needs to be repeated. Right, so my final example brings in a bit of systematics. I've always been interested in the interface between ecology and systematics, and I want to show an example of where the study of ecotypes can lead to decisions about classification. This is a spectacular orchid, one of the largest in South Africa, found all the way from the Eastern Cape through the Drakensberg Mountains into KwaZulu-Natal. And in his revision of the genus Diza, Peter Linder felt that although there appeared to be several forms, that they formed clinal variation and could not be recognized taxonomically. So that was his figure over there. So he felt that the variation formed a continuum. Botanical artists writing flower guides obviously didn't agree, because in several books they've illustrated both forms. So here's the lowland form, illustrated in both forms. There's the what we call the Montaigne form, and there's the Eastern Cape form, also what we call the Hogsback form. So I tried to persuade my colleague, Benny Betebeer, that we should address the systematics of this situation, and he wasn't convinced. He felt that Peter Linder was probably right. But when I looked at the scent chemistry, it was clear that the high altitude one had almost no scent, whereas the lower elevation forms were strongly scented, suggesting that there may be a pollinator difference. And it's incredibly hard to document the pollination systems of these plants. The lowland one, after many years, I established was pollinated by hawk moths. They carry these huge pollinaria underneath their thorax. I've highlighted in yellow over there. This is showing one in action. And as the moth comes around, look at the massive mass of pollinia hanging from the thorax of the hawk moth. Now, the pollination of the high-altitude form was more of a problem. This involves a day hike to get to a population and camping at a site. And I'd struggled for years. I'd seen a few pollinators, long proboscis flies, on this form. And eventually I was going to Tanzania earlier this year, and I asked Ruth Cozy, a postdoc in my lab, who's one of South Africa's top trail runners, if she wanted to get some exercise in the Drakensberg, and she readily agreed and went up over a period of several weeks, dropping off camera traps at these sites. And she really made the important breakthroughs in determining the pollinator of the unscented or weakly scented high elevation form. So it's pollinated by this long proboscis fly, Brassica gangobaria, which we've seen in some of the other systems as well. Here I've just taken one of the videos and slowed it down so you can see just how extraordinary this interaction is. And this fly does eventually find its way into the flower. So in this case, after 1,000 camera hours, she documented 20 interactions involving pollinators. This means that each plant, and these plants are quite sparsely distributed, is visited on average about once a week. So you can see the formidable challenge in documenting the natural history of this system. So again, the mountain form is associated with this high altitude fly, and the lowland form is associated with hawk moths, which occur at lower elevations. The morphology, this is a non-metric, multi-dimensional scaling plot of morphology. You can see the difficulty, even Peter Linder had observed, separating these two forms, the Eastern Cape and the lowland form. But if you look at the scent chemistry, the actual scent composition is completely different between these two scented forms. The orange over here and the green. This one has no phenylpropanoids. This one has a whole lot of eugenol and various derivatives. So this eventually convinced my systematist colleague that we should do something about it. So he put together this phylogeny, and here came the big surprise, because these morphotypes, we threw in some closely related species. Now remember, Peter Linder decided that you couldn't separate this into more than one species. In fact, two other related species turned out to be nested within what we thought was a set of morphotypes. And here we had no choice but to split Diza crassicornis, this is the type species, here into 3 taxa. That's an old name we resurrected, and this is a new name we're proposing. So in this case, we made the obvious decision to change the taxonomy. Now, just to finish off, and here it gets more controversial, I've used the word species level in this talk as if I know what the species level is. And as you know, this has been a source of intense debate over the past 50 years. What do we actually mean by species? And there have been numerous species concepts proposed. Some people, like Doug Shemsky, whose work I greatly admire, have said, well, let's just settle on the biological species concept and we'll only allow one new species concept every 100 years, which I don't think is the answer, because I don't think you can solve a complex problem in biology just by simplifying the definitions. But I've come to believe that all of the major species concepts express something true about the nature and identity of species, although not all of them are applicable in all cases. And I don't need to remind you why each of these properties of species why they apply to most species, but not to all species. So the question then is, are ecotypes, as Vern Grant suggested, on the way to becoming species? Or are ecotypes simply divergence that didn't result in new species and which will simply sort of have short-lived evolutionary trajectories? So I wanted to just point out an example of how problematic this is. In the last 5 years in current biology, there's been a big debate about how many giraffe species you'd have in Africa. You couldn't think that there'd be a more obvious and simple species delimitation problem than giraffes. But giraffes have disjunctions in Africa. They consist of different genetic clusters, and some people have been proposed that, well, if you have these separate genetic clusters, you must have separate species. So we'll have 4 giraffe species with a couple of subspecies thrown in. Other zoological taxonomists have said there's only 1 giraffe species in Africa. The same debate has occurred over forest elephant versus savanna elephants in Africa. These are big, spectacular mammals, and people can't agree about it. So you can see why it's an endless source of debate for plants. So this is going back to plants. This is how I see lineage diversification in plants. Usually starts with some kind of geographical disjunction, adaptation to habitats, perhaps flowering time thrown in, a pollinator shift, and eventually after some time the development of genetic incompatibilities. But the order of these events, of course, can vary for any particular system. If it was allopolyploidy, for example, the genetic incompatibility may come right at the very beginning. The point is there's no sort of it's not a one-size-fits-all view that one has of speciation in plants. And generally, ecogeographic isolation is much more important than pollinator isolation in plants. This is a very helpful paper that I recommend to people to read. It's a paper by Resnick and Rickliffs, who pointed out that Darwin's conceptualization of how microevolution, for example, ecotype formation, relates to macroevolution, was a process of continual small divergence events combined with extinction to give you this macroevolutionary pattern of species with strong phenotypic or genetic discontinuities between them. And I find this a very helpful way of thinking about the evolutionary process. So I've just sketched it out as you might think this is how most people think about lineages. This is how you think about lineages with extinctions. This is how Darwin thought about lineages. All these sort of ecotype kind of experimentation within lineages, going extinct, forming, and this resulting in the diversity that we have today. So I think for people working on other plants, an interesting challenge is to see how macroevolution correlates with microevolutionary pattern. In Laperosia, for example, there have been many pollinator shifts, and below the species level, you see some of these pollinator shifts as well. In general, for plants, if there have been ecological shifts at the macroevolutionary scale, you see those same ecological shifts happening at the microevolutionary scale, suggesting that the microevolutionary process is the same one that gave rise to the broader macroevolutionary pattern. So these are 2 species which consist of ecotypes in Laperosia. So to sum up, just to remind you, these were the species, the systems that I looked at, each having different properties, different attributes. And just as an agenda for future research, I just want to highlight these 6 major problems that I identify here. When I started working on sort of evolutionary diversification in the 1990s, phylogenies were the rate-limiting step. Now phylogenies are widely available, and for many plant lineages, the rate-limiting step for understanding evolution is actually natural history. We simply don't know enough about the interactions of plants in order to be able to interpret the function of traits. There's a strong absence of data on ecological gradients. I think we've done quite well in South Africa in identifying these ecological gradients. There's very little data elsewhere in the world that's similar. Practically, the design and the interpretation of reciprocal translocation experiments is incredibly challenging when it comes to flowers. Optimizing the direction and the number of times that shifts occur on phylogenies becomes very difficult, particularly when there are numerous shifts on the tips. It becomes very difficult to reconstruct ancestral conditions and to work out the direction of shifts. So it works well when you have just a few states at the tip. It becomes much more complicated when there are a lot of states at the tips. The problem of if you have divergent allopatric population clusters, how do you know if they should be recognized as separate species or not? There's simply no answer to that problem. In fact, even Wallace identified this as a problem in his early work in working on sort of island archipelagos. And finally, are ecotypes the process, or are they a step in species formation, or are they simply just short-lived experiments? I think the answer to that question, I would say, is that most ecotypes probably don't give rise to species. They probably eventually become extinct. But I think most species probably arise originally as ecotypes. That would be my answer to that question. And then finally, just some people to thank. Yeah, so thanks for listening.
Okay, thanks, Steve. I'm sorry that we have not time for questions. Then if you want to ask, we have more days to speak with Steve. Thanks again.